that at the gills and at the level of the kidneys. Okay? All right. Let's take a look at freshwater fish. Freshwater fish. Should have left my fish diagram up there. All right, there's my freshwater fish here. Now my problem is I'm hyperosmotic. And fresh water doesn't contain a lot of dissolved substances in it like seawater does. So the fresh water here is hypoosmotic. Now, hyperosmotic, high concentration of salts or active molecules that actively participate in osmoregulation. They're not osmoregulation. I told you it's going to be one of those days. Osmotically active, and that means that water is being pulled into the fish. Okay? Now, the place where most of this is going to happen, again, is these gills. Right here is where most of the water is going to enter into the fish. I can't provide a very good barrier to the movement of water here. So water is coming in through these gills, right, all the time. Water is coming in through the gills all the time. Now, the problem for the fish is as the water comes in, it alters the concentration of water in cells. It changes the concentration of ions, because as I add more water, I decrease the concentration of ions. Those ions need to be at particular concentrations. I got to get rid of the water. So one of the ways to get rid of the water is to just go to the bathroom all the time, right? The kidneys are laboring all the time, and what they're basically doing is producing this extremely dilute urine, and it's just peeing it out all the time. Very dilute, copious means large amounts, of urine containing very little salts. In the seawater fish, the kidneys working overtime to get rid of salts and keep the water. In a freshwater fish, the kidneys are working overtime to keep the salts and get rid of the water. Okay? All right, now I can also here at these the gill epithelia is to try to take up any ions that I can find out here in the environment. There's not very many of them, but they're there, is to try to take these up as much as I possibly can. Is that okay? So I'm going to take up these salts, the active uptake, that's active transport, of sodium and chloride across the gill epithelia. Okay, water's coming in and going out. And then I can also get some salt and water from my food, and depending on what I'm eating, I can either get a higher or lower concentration of water and salt. And notice the difference here is that these freshwater fish are not drinking water, whereas the saltwater fish are drinking the water in all the time and then trying to get rid of the salts all the time. Okay? All right, so that's the bony fish. Okay. Let's move to some terrestrial organisms. Start with a bird here. This particular one is a penguin. Okay. All right, now, the reason they've gone with a penguin for this example is that it's a seabird. It feeds on a lot of organisms that live in the ocean. And because of that, it's taking in different concentrations of salt water, water and salts. If this seabird feeds on sea stars, it's getting water at the concentration of seawater. Right? If it feeds on more fish, it's getting less salt than it would if it fed on sea stars. Okay? All right, so let's take a look here at this particular configuration here. This is part of what's called a salt gland that occurs in fish and some reptiles. So what we've got here is blood. This would be capillary system. You can see that capillary system there. And then here what I have is some interstitial fluid. It's fluid between cells, right? It's not cellular fluid, it's between cells. This is fluid that lies between the capillary epithelium, and here's another epithelial layer here. Okay? So I've got capillary, that's capillary, and this is an epithelial layer. Okay? Now, what happens here is there's some sodium chloride diffusing from the blood to the interstitial fluid. The concentration of salts in the blood is much higher than it is in the interstitial fluid. These salts have come from the digestive system, right, when I consume things, and the salt is going to diffuse in. Then we've got these epithelial cells here that are going to do active transport to move the salt into this collecting duct. Okay? And the idea is that the penguin is running its blood across these collecting ducts. The salt diffuses out of the circulatory system into the interstitial space, and then you invest lots of ATP to actively transport these salts into the collecting duct. And as you move salts into all these different collecting ducts, this salt begins to collect in all of these secretory tubules. And these tubules all come together until we reach a very specific spot here. And then what we have is salt being pushed outside of the body onto the surface of this bird right next to the salt glands. Okay? So the seabird is getting salt in its diet, right? The only source of water is seawater. They could drink the seawater, but again, it's too much salt. Their bodies are hypoosmotic relative to hyperosmotic seawater. They need to get a way to get rid of this salt. Here, we're doing an excretory function at these salt glands to excrete the salt outside the body. We have to invest energy to do it, but that allows us to keep the water but get rid of the salt. Okay? So I'm going to go ahead and take a break right here, and when we come back, we'll do some more excretory system.